purpose of this discussion is just to give you kind of a walkthrough from two papers that I've chosen. These are not required reading, but I wanted to give you an example of how some of the principles that we've talked about are applied in real life studies. So this is a primary study. They gathered data. They're looking at population structure of savanna elephants in Kenya. And I'm just going to highlight some things that stood out to me. There's a lot of good stuff in here. It's fairly approachable for a research paper, but as long as you understand the principles that I'm going over, you'll be fine. If you'd like to get into it in a little more detail, if it interests you, fine, go ahead and read it in a little bit more detail. Whenever you're reading a paper, the very first thing to do, of course, is go over the abstract. It gives you an overview if it's written well. Not all abstracts are, but it should give you an overview of the structure of the research, how it was organized, and also what their results were. Okay, so they're looking for genetic structure among these savanna elephants in Kenya. They're using two different types of DNA. They're using one locus is the singular, one locus in the mitochondria. They're looking at a hypervariable region in the control region of the mitochondria. And then they're looking at microsatellite. I think they had something like 20 different microsatellite locations that they're looking at. So microsatellite locations are all nuclear, and of course the hypervariable region, the mitochondria, is a mitochondrial marker. Okay, and then you'll see a lot of F statistics here. We're only going to look at the FST, but realize all of these are different types where they're basically comparing different levels of genetic diversity and determining whether there is a significant difference, for instance, between one population and another, or, as they've done here, between one region and another. So they have, I think, 8 different populations that they've studied, and they've divided these 8 populations into 3 regions, kind of larger areas that each contain more than one population. Okay? And then finally, they're looking at if there's any significant difference between the data they got from the mitochondrial area and the data they got from the nuclear areas that they were looking at. Okay, and then lastly, the results, they have 3 broad regional mitochondrial groups, so that fell well into their geographically, and they observed that in the population genetics and in the geography. And then there are some thoughts which are not really relevant for this class, but this applies to ecology and conservation. So that really shows how population genetics and phylogenetics are really applicable across all disciplines of biology: ecology, conservation, morphology, human medicine, epidemiology, you name it. And these methods really can play a really important vital role. Okay, so introduction gives a lot of background about elephants, about their historical population pressures, about some of the recent comebacks that they've had, and then other studies that have looked at either elephants or other organisms that may have unexpected have a similar population structure. This region talks about how they amplified it. Now, I want to say one quick thing about sampling. When you are sampling DNA, you really need a large sample size. If you want to get an accurate picture of genetic diversity, you have to have a large enough sample size to have a reasonable chance of getting an accurate picture of the diversity. For my undergraduate classes, I always use the analogy of a political poll, and that works well during this election season. If you only ask 10 people, there's likely to be strong bias just by random chance. You maybe can't even predict one way or the other if you're randomly picking 10 people from the U.S. population. Even worse, if you have too small population size and it is concentrated in one area. So let's say we asked everybody in Austin, or even 100 people in Austin. Reasonably, we couldn't ask everybody, but we ask 100 people in Austin. That might not tell us much about the state's political preferences or the nation's political preferences. And so when we do political polls or DNA sampling, we want a large enough, and hopefully randomly across the entire population, but a large enough sample that we have a good chance of capturing an accurate picture of what's going on. And then, of course, your statistics are dependent on the sample size. So your certainty, your whether or not things are significantly different or not, you're going to have more power to determine that if you have larger sample sizes. Okay, so again, they're doing 8 populations. They divided them up into 3 different regions based on geography. Okay, so I think they have the south region, the middle region, and then the, let's see, where are the regions here? I guess that's down later on when they do the analysis, okay? But anyway, 3 different regions. And again, 2 areas, mitochondrial DNA, some details about how they got that data, not critical for our studies. And then the microsatellite and how they did that. And there's some information specific to microsatellites and how you have to do a little bit of statistics and control for that, right? So here is a map of where the different samples were. So notice some of these may be a little undersampled. If you only have 8, 9 individuals, have you really captured all of the genetic diversity from that region? Maybe not, but that's where the statistics come in as you're doing the analysis, okay? And then they do pairwise comparisons. Look at this is just a lot of numbers, and unless you're an expert in this area, you may not know it, but they've got the ones that are significant, the ones that are highly significant. So you can see some structure between the 8 different areas, okay? And then they go over the results. So if you are really interested in a paper, or if it's assigned for a class, you may need to spend a little bit of time on results. In this case, we'll just go over it quickly. So they're looking at the 159 base pair mitochondrial control region. They're talking about how the overall nucleotide diversity is fairly low. In some areas, it's extremely low. There might only be 2 different haplotypes in that area. And then they're looking at tests, these FST tests, looking between different areas. I think they do both a regional comparison and a population comparison, okay? They go in the microsatellite information and some stuff about the statistical analyses, not really critical, but they do show some significant structure between the different regions. And then the last thing that I want to point out is kind of an interesting thing here. They see something different, and maybe something that they even expected, between mitochondrial homogeneity tests. So the differences between areas based on mitochondrial genes were fairly low, but the difference between areas based on nuclear genes, most of those comparisons were much higher. And there's a reason for that based on elephant social structures. So in elephants, there's a matrilineal society where females will often stay with their mothers and sometimes even take over, and there's a single female that is kind of the matriarch or the leader of a group of elephants in an area. And males, once they hit puberty, typically have much more pressure and almost always will get up and leave and wander around. And then for the rest of their life, they also usually have larger ranges. They will stay with one group and then join another one, and so they're not as set in the social structure as the females are. And so what this means is the mitochondria, which can only move through the mothers, right, is going to be less diverse because we have less spreading of maternal genes because there's just less movement of the females. And the males, which are moving around, so the nuclear diversity is controlled by both males and females. And because males are moving around much more than females, we see that discrepancy between the nuclear genome and the mitochondrial genome. If males and females were moving around equally, then we would expect to see roughly similar levels of diversity between mitochondrial markers and between the nuclear markers. So that's kind of an interesting thing that reflects the structure of those organisms. And then finally, we won't get into this, but there is some information about how this might help conserve and manage and things people need to be aware of based on this genetic diversity. The second example of a population genetic study that I've chosen to look at, it's a little bit dated, but this is kind of a classic study and a classic example of how population genetics can tell us something about the structure of a population and also its recent history. So it's been known for a long time that there was a fairly recent bottleneck in the population of cheetahs, African cheetahs especially. Now, there are cheetahs that are historically have, and still to this day, are in the Middle East, and I think a little bit up into southern Asia. But those populations are under stress also. But in Africa, there was a very clear bottleneck fairly recently. We're talking tens of thousands of years. And it's estimated to be at the late Pleistocene when there were other major near extinction events. And the reason for this is not because we have fossil evidence or historical records or anything like that, But the reason is because the cheetah population, although it has rebounded since that near extinction event, their genetic diversity has not returned. So quickly we'll look at the data, and then and hopefully you're familiar with this idea of a genetic bottleneck and why it causes loss of genetic diversity across the entire genome. Whether or not there is strong adaptive advantage, natural selection working on a site, or whether the site is completely neutral, genetic diversity is just dramatically lost during a bottleneck event. So a couple things, again, abstract, take a look at it, but they're using the same types of markers. Now, their data is not quite as large. This is a large data set for the time, but they were limited more in their ability and how they did it. So they're using restriction length fragment polymorphisms, electroallozymes they're talking about. And so they are looking at these microsatellite loci and how long they are, right? So basically just counting how many repeats they have. And then they're also looking at mitochondrial DNA. So they're looking at both of them. And so let's talk briefly about what they say here, and then I'm going to give you an overview of a review, hopefully, of population bottlenecks and their impact on population. So it talks historically about how there was this ice age and perhaps some human pressure that caused major population crises in many species, and the cheetah nearly went extinct. So they say a 90 to 90% reduction in allelic variation, meaning that probably only 1 to 10% of the historical population survived through this crisis event. They were able to survive, things got better, and then they were able to expand again and fill back up much of their range, but without their previous genetic diversity. Okay, so they're calling this a demographic contraction or bottleneck. Okay, now this has some impact on many genes that need to be variable to be very healthy. So they're talking about spermatozoa, infant mortality, MHC class 1 genes, which are involved in mammalian immune responses, and the more diverse they are, the more robust your immune system is. Okay, so it looks like they're actually they found 2 bottlenecks. They're trying to time these out, and this is based on the amount of diversity they see in a population of a certain size versus the amount of diversity that they would expect. And the reason for this is because of this phenomenon of the bottleneck. So here is an example with marbles representing alleles in a population. And at a bottleneck, if you subsample it just by chance, you're going to oversample some of these alleles and undersample others. So they've only let 5 alleles, maybe 10% or less, right, come through this bottleneck. And notice we've immediately lost one of the 3 alleles, and the other allele is represented at a much lower frequency than it was before. And so it's going to be subject to loss due to genetic drift, particularly if it takes a while for the population to rebound, right? So bottlenecks have this kind of trifecta of impact. Number 1 is there is an initial loss of diversity, and that's fairly severe. Number 2, if the population remains small for a while and it takes a while to rebound, we're going to have strong random fixation of alleles across all loci. And we're going to lose more alleles than were even lost in the initial bottleneck due to fluctuation of allele frequencies by genetic drift. And then finally, we also have inbreeding depression. If we have more higher frequency for recessive alleles that are dangerous, maybe they're at a very low frequency, but some of them get represented more, that we're going to see more population pressure based on what is called inbreeding depression. And that's when you mate with close relatives, you have more likely to pair your few rare recessive alleles. Your offspring are going to feel the consequences of that. So it's kind of this perfect storm, you will, or trifecta of results based on this recent bottleneck effect. And the cheetahs have recovered population-wise, but it's going to take them many tens of thousands of years and new mutations occurring and new genetic diversity build up to get back to the levels of diversity that they enjoyed prior to this bottleneck event. And so during that time, they're going to be more fragile and more susceptible to extinction events. So even today, cheetahs are more of a concern, even though their populations are fairly healthy compared to many other endangered species. They are more of a concern because they have smaller levels of genetic diversity. Okay, so they're looking at microsatellite loci, but rather than doing actual DNA sequencing, this is one of these older methods we talked about, they are using restriction enzymes to break up DNA and then see how many pieces it breaks into. And then they're able to categorize different alleles based on how many bands show up in their results. So it's a less precise way because you can't see every single change, but you can catch some of the changes and begin to capture a big picture, at least, of the diversity. Now, notice they had to use 28 different restriction enzymes to be able to find the few that gave different patterns. So it was a very labor-intensive way to do it, and today we can do it much more easily by just sequencing the entire area, and that way we can, if we sequence and get good sequence, we can identify every single genetic differentiation, every polymorphism present in that population. Okay, and so they look at it, they do these analyses, they show you some of the results, they make a gene phylogeny based on this electrophoresis. So this is going to be using a distance method, right? So remember we talked about distance methods are not used largely today, but they were used in the past, okay? And it looks like they have a distance and a parsimony one there. So they, more information. This is a lot of details. You don't need to get into this. But then they start looking at these nucleotides. And so basically what they say is they confirm the values for how much genetic variation is present in the populations, based on felines of similar population size, other species. And so they say about 10,000 years ago, there was a major bottleneck event for the cheetahs. And they've also been able to even capture some of the regional stuff. And there have been more papers that have used larger data sets to confirm some of this. But there's been a more recent geographic partition of the eastern African ones and then the southern ones. These are subspecies, different populations. And so they notice some distinct differences. So there are clear genetic separation between those two populations. Okay, and then of course this has some impact on conservation and captive breeding programs, and how you might choose cheetahs to mate with one another to try to preserve what little bit of genetic variety is present in that species.